Hello, my name is Stewart Fisher, and I'd like to welcome you to the Mastering Medicinal Chemistry Conference, which will be held May 21st and 22nd at the West End Waterfront Hotel in Boston this spring. This exciting conference will cover a number of hot topics of interest to medicinal chemists, including allosteric inhibition, natural product chemistries, covalent inhibition strategies, and the role of binding kinetics in drug discovery. On a broader perspective, the use of covalent inhibition to enhance pharmacological impacts is closely related to the growing interest in slow binding kinetics in drug discovery. I think that the role of slow binding kinetics has reemerged as one of the most important concepts for small molecule drug discovery. In the past, drug discovery programs focused almost exclusively on the use of thermodynamic measures of binding potency to select and advance candidate molecules, despite the fact that at least 1/3 of the marketed drugs utilize slow binding kinetics in either their mode of action or improved therapeutic window. The realization that kinetic profiles present a distinct and orthogonal measure of compound efficacy has become more widespread over the past 5 years and is now seen as a critical, but still largely unexploited area of analog design. This conference has numerous opportunities to better understand the integral role that kinetics, along with thermodynamic measurements, play in effective lead discovery. In the first instance, I will chair a case study session on inhaled p38 kinase inhibitors that will be described by Dr. John Matthias, where slow binding kinetics, along with formulations required for inhalation administration, were critical to success. Second, the latter half of the conference will highlight 4 talks on this subject from experts in the field, including Professor Peter Tong, who will describe the linkage between target vulnerability and extended residence times, Dr. Rick Roberts of Almirall to describe the impacts of extended residence time in the discovery of potent CRT-H2 antagonists, and I expect that my former colleague, Dr. Anne Boriak-Shadin from Epizyme, will give an excellent talk on the rational design and development of potent selective inhibitors of the DOT1L histone methyltransferase. I will also present in this session and will focus on the challenges facing translating in vitro measures of slow binding to cellular and in vivo disease models, and demonstrate the power of predictive mathematical models that utilize in vitro kinetic parameters. Historically, empirical correlations have been used to track the effects of in vitro kinetics on cellular and in vivo efficacy. I think we can do much better in the future. To ensure that there is sufficient time for discussion and debate, I will be chairing a discussion on this topic during the conference as well. Finally, for those that are interested in getting more background on this topic, Professor Peter Tong and I will co-teach a pre-conference short course on this topic. This course will cover all of the core concepts underpinning binding kinetics and their application to in vivo systems, and will utilize a number of recent real-world examples to demonstrate the impact and application of these concepts. I think this conference will be an excellent venue for networking across a variety of traditional boundaries, across disease area specializations, and within and outside the academic and industrial environments. I expect the conference to be a vibrant, intimate, and fully accessible to all participants from the exhibit hall to the multiple poster sessions. As a result, I'm looking forward to discussing leading science, meeting new contexts, and generating new ideas and having fun in Boston in the spring. I look forward to seeing you in May.